And in 2015 and 2016 and 2018, he was identified by Thomson and Reuters as a highly cited researcher in the discipline of pharmacology and toxicology. He has supervised 67 PhD completions and supervised over 20 postdoctoral research assistants. Currently, he is supervising a team of 16 PhD students and research assistants. So Professor Pouton contributes to the research field of nucleic acid delivery, stem cells technology, drug delivery, and vaccine designs. So in relation to COVID-19 research, he has been using his experience in nucleic acid delivery to design and deliver candidate mRNA COVID-19 vaccines. So it's my honor to invite you to deliver your presentation, Professor Pouton. Thank you very much.
Thank you very much for that introduction, Rohini, and it's an absolute pleasure to be with you all today. I might, just to make sure that the video link is, the lecture can be heard properly, I might turn off my video link just in case the bandwidth causes some problems with viewing the slides. So I'll come back a little later. So let me share the slides. Well, the year 2020 and carrying on now into 2021 is not a year that any of us will forget in a hurry. And obviously, it's been an extraordinary year for all of us, and in particular for the understanding of the science of vaccination. And so I'll take you through some of the developments and some of the reasons why we, in a way, succeeded in producing vaccines so quickly against COVID-19, but I'll also illustrate that the problem is not over yet. And whilst we need to vaccinate large populations of the world, we also have to face the prospect of variant viruses which are emerging and think about what we might do about that. So I'll just start by saying that it was remarkable around the beginning of December how one vaccine, the BioNTech Pfizer vaccine, was already approved for human use. So the New York Times has been following the development of vaccines quite closely, and it's a good source of information about how many vaccines are in development. And this was the slide. This is a slide taken from the New York Times in December 2020. And you can see that this was just after the first vaccine was approved. But you can also see that there are a lot of other vaccines. There were a lot of other vaccines in development at this stage as well. Some 40 vaccines in the phase 1 of clinical trial, another 17 in phase 2, 14 in phase 3, and some other vaccines that had been approved for early use or emergency use. Just a few months later, in April, the situation had changed again, and now we had 4 vaccines had already reached the stage where they'd proven to be unsuccessful. But we had 8 vaccines approved at this stage and several others in development, and that continues. So at the moment, we're in a position where there are many other vaccines that haven't come through yet, which may contribute to the overall vaccination program we need to succeed. The interesting thing about these vaccines and how quickly they developed was that most of the early vaccines were developed in so much more rapidly than normally takes place with a vaccine development. And there are a few reasons for that, which we can talk about. So generally, to develop a vaccine, there's a period of preclinical testing, which usually means testing in animals, possibly ultimately in a primate, before going into human studies in the clinic. And the human studies normally take place in a safety study at phase 1, a smaller efficacy study at phase 2, and then a large efficacy study at phase 3. And then they go into general use, and then monitoring is sometimes referred to as phase 4 of the clinical study, post-marketing surveillance. Normally, the timeline for the development of any vaccine really is quite a long period in development, maybe in excess of 5 years, and then quite often the clinical trial program is as much as 9 years. But obviously, in the wake of the pandemic, an extraordinary acceleration has occurred in the development of vaccines, so that really the clinical program was only 6 to 9 months. And Moderna, the first group to get into humans in a phase 1 clinical trial, started their phase 1 clinical trial just 60 days after the genetic sequence of the vaccine was released. So 5 to 10 years was compressed into 60 days in the pandemic, which is quite extraordinary. Now, what's the reason for that? Well, one of the reasons is that we have these new technologies, one of which is the mRNA vaccines, and I'll go into more detail about that. But also, there were also developments in DNA vaccination, and the viral vectors that we hear about, like the AstraZeneca Oxford vaccine and the J&J vaccine, are essentially DNA vaccines, but delivered using a viral vector. So whilst this DNA vaccination program has not really progressed very far, we've seen that the ones that are actually approved are either mRNA vaccines or viral vaccines based on adenoviruses, and there are a number of those that are already in development, also in China, and also the Russian vaccine is also an adenoviral vaccine. So what makes these technologies special is that once you've got the sequence of the genome of the virus, you can very quickly produce a viral candidate, a vaccine candidate. And so, and we were doing this ourselves in our laboratory too, so that it only took maybe 2 weeks to go from seeing the sequence of the viral genome itself to going to an mRNA vaccine. So probably Moderna were able to do that in maybe a week. And then we're starting animal experiments to see whether the vaccine worked. And also safety experiments, toxicology studies, so that they could get all through that process within 60 days. Now, these vaccines are much more rapid to develop than a traditional vaccine. But the traditional vaccines based on either proteins or inactivated viruses are still emerging for COVID-19 and may have an important role to play going forward. We just don't know at the moment. There are so many unknowns in this pandemic. Let's just take a little look at the virus itself before we progress to vaccines. So this is a schematic representation of the genome of the virus. And the virus itself looks fairly simple at the outset because there are only 4 proteins. There's a package of RNA and then only 4 proteins, one of which is the spike, one of which is the condensation protein, which condenses the RNA. And then we have the membrane protein and the envelope protein. So really only 3 viral envelope proteins, and then this RNA condensing protein. But actually, there are many other proteins that this virus produces. So it's actually a very complex virus and quite a big one. Its genome is quite large, and it has a whole load of ways of interfering with our cell functions and our immune functions, which are not fully understood. So these non-structural proteins, which are encoded by a large chunk of the viral genome, are not really fully understood in their function, but there are some 30 different proteins which are likely to be produced in mammalian cells. And then there are the structural proteins which make up the viral capsid itself, and some accessory proteins which are involved in assembling the virus. So we've got something which, on the face of it, if you look at the viral particle, looks quite simple, but there's actually a huge amount of complexity built into the life cycle of this virus. And there's a lot to be learned about it. And it may well be that we need to adapt and modify vaccines to overcome some of the ways in which the cell's machinery is interfered with by the virus. What made it possible to get to the viral vaccine, to get to the vaccines very quickly, was an understanding of other coronaviruses. So I'm sure you all know that COVID-19 is caused by a virus we call SARS-CoV-2, but the SARS virus already was responsible for an outbreak in the early 2000s. And that outbreak led to quite a strong understanding of coronavirus biology. And it turns out that the SARS-CoV-2 virus has strong homology with the original SARS virus. And that meant that there was a good understanding of the structure of the spike protein. So the spike protein has a structure very similar to the original SARS virus. And that understanding of the structural biology allowed scientists to compare the structure of the SARS-CoV-2 spike with the original SARS virus, and very quickly build a structural model of that virus, which was then confirmed very quickly using cryo-TEM technology, which again is a technology which has not been around very long for finding crystal structures of proteins, and that was used within a month of the viral genome being produced to have a structural model in great detail of the SARS-CoV-2 spike. So that itself was a remarkable piece of science, but it allowed us to be aware that like the SARS virus itself, this is a trimeric protein, meaning that the spike itself is made up of 3 identical fragments which fold into this structure. And there are 3 binding domains, which we call the receptor binding domain at the tip of the spike. So those small fragments are the ones that actually interact with the receptor on the surface of our cells, the ACE2 receptor, and it's that which we're trying to raise antibodies against this tip. And that's an important point, because when you look at the whole spike protein here, you'll see that there's a huge amount of protein in the extracellular environment which does not bind directly to the receptor. And most of the vaccines that we have at the moment, with the exception of one which is not fully available throughout the world, most of them are vaccines against the entire spike protein. And that's a very important thing to take in, because it has big implications to how these vaccines will work and whether they're optimized. And I'll come back to that. So the virus binds to the ACE2 molecule, which is present on endothelial and a lot of epithelial surfaces, and obviously the virus infects via the lung through this ACE2 receptor predominantly. It gets taken into the endosome of the cell and then fuses with the endosomal membrane, and the viral contents are released. So what are the opportunities for COVID-19 vaccination then? And I'm going to focus particularly on the scope for design of second generation vaccines, because we all know that we have quite effective vaccines already against the whole spike, but they're also vaccines against the whole spike of the original isolate, which is what we often call the Wuhan strain of the virus. you can see here a schematic of the genome again. You can see this in green. This is the receptor binding domain, which is encoded by this piece of protein at the top of the spike. So the idea of these vaccines that are already approved for human use is to generate antibodies against this spike receptor binding domain. The problem with using the whole spike is that there are lots of epitopes around the other regions of the spike protein, which will also induce antibodies to be raised. And the antibodies that bind to the other components of the spike are likely to be non-neutralizing antibodies, meaning that they don't bind to the RBD region, and therefore they don't really prevent infection. The problem with that is that having antibodies against other components of the spike can cause problems. It can cause a phenomenon known as antibody-dependent enhancement, which we know from SARS can make the virus more infective. Now, we haven't seen that for COVID-19 yet. And one of the reasons probably is that COVID-19 already has a very high infection rate. So perhaps this enhanced antibody effect, which was seen with SARS and also with MERS, the Middle Eastern variant of coronavirus, maybe that doesn't happen with COVID-19 because it's already such a strong binder to its receptor. But there's also something which occurs, which is associated with the more problematic disease that occurs in COVID-19, and that is this enhanced respiratory disease. So a lot of the viruses that are non-neutralizing are thought to be taken up because they coat the virus with antibodies with their Fc domain. They're thought to be taken up by macrophages, which can then traffic to other regions of the body, deep into the lung, maybe into heart tissue and so on, where we see complications with COVID-19. And that can lead to the delivery effectively of intact virus to deeper regions of tissue rather than just the initial airways. So it's probable that some people that suffer really badly from COVID-19 are suffering badly because they excuse me a moment.
Hello, Professor Pouton. We can't hear you. Can you please unmute your mic?
Sorry. I saw the message. I didn't quite understand what it meant there. Yeah, I don't know why suddenly I was muted, but okay. Hopefully you can hear me now.
Yes, we can hear you now.
Okay, good. Yeah, so if we use the receptor binding domain alone, then it's quite possible that we can raise antibodies which are neutralizing and don't cause these other problems. And that might effectively be a better way of vaccinating the elderly people that suffer badly from COVID-19. So you have to remember that with vaccination, we have really two objectives. We have the objective of reducing the spread of COVID-19, and that really means inoculating large proportion of the population, but we also have the objective of protecting people from serious disease. And one of the problems with using the whole spike vaccines is that they may be quite good at vaccinating naive individuals, but in individuals who've seen a lot of coronavirus infections before, and I don't mean SARS or MERS, but just the kind of coronaviruses that cause common colds, it may be that they're raising antibodies against regions of the spike from other coronaviruses and it makes them primed to keep on generating these non-neutralizing antibodies rather than generating new antibodies against the spike. Whereas a naive individual, a young person who is vaccinated, probably sees this RBD and raises some good antibodies. So I think this is one of the interesting things as the antibody program develops. We may find that we need to think about which vaccine to choose to protect people who may be vulnerable to COVID-19, and maybe that we need a way of detecting individuals who are already primed to make non-neutralizing antibodies. Another thing to bear in mind is that all of the vaccines so far have really been aimed at making antibodies against the spike, and they sort of have ignored, to a large extent, the T cell responses that might also play an important part in the immune response. And we really don't know too much yet about how the vaccines induce T cell responses and which ones are useful. So there may be improved vaccines in the future, which maybe we need T cell responses against other proteins within the viral genome itself, rather than just the spike protein. So I think that is something which gradually will emerge as more research is done. One of the things that has also been remarkable about the science surrounding the COVID-19 pandemic has been how rapidly tests have been developed to test and find these antibodies, and also to develop cell culture models of infection. So very quickly, scientific suppliers were supplying antibodies, ELISA tests, and surrogate neutralization tests that scientists around the world could use. And that's been really valuable to be able to buy those things off the shelf and use them to assess the quality of immune response. Locally, my own group have been able to collaborate with a group at the University of Melbourne at the Doherty Institute, where they have PC3 containment facilities at the Doherty, and they're able to actually run an infection model using mammalian cells and ask the question, can we prevent with our antibodies, our anti-sera from mice experiments, for example, can we prevent infection of mammalian cells with the serum samples from mice? And that's been a very important component of our study going forward. And I'll show you some data a little bit later. The other thing that people have been doing, which is a very interesting complement to these two assays here, is to take serum samples from patients who've had COVID-19 and ask the question, how does our vaccine serum correspond and how does it compare with serum from patients who've actually had COVID-19 and are recovering from it and have immunity? This is just an illustration of the problem that I was referring to before, that if you raise non-neutralizing antibodies, what happens? So a neutralizing antibody against the spike protein will prevent the binding to ACE2. But if you produce non-neutralizing antibodies, the outcome is usually uptake by recognition of the FC tails of antibodies by phagocytic cells. And that can lead to stimulation of cytokine release, which can be problematic. So one of the things that occurs in serious cases of COVID-19 is that you get these pro-inflammatory cytokines being produced, which creates potentially a serious inflammatory condition in the lung. So I'll turn now to the issue of mRNA vaccines. What's the advantage of mRNA? Why did they get produced so quickly? And why are people so interested in mRNA rather than any other form of vaccination? Well, it's something that can be produced quickly and in a very pharmaceutical manner, because mRNA is produced without any, it's a cell-free process. You have to produce some DNA, which is usually produced from bacteria, but once you've got the DNA and purified it, you can produce mRNA in basically just a stirred vessel. It's just an in vitro technique using bacterial enzymes. So you need a polymerase and you need nucleotides. You stir them up in a pot and then you purify the mRNA. And the mRNA is a compound that can be purified by chromatography. So it has really strong advantages from a pharmaceutical point of view. It's a very pure material. It looks something like this schematically. So you have a coding region of RNA. It also has some other features that are quite important that you have to include. Usually there's some untranslated regions which are quite important to maintain and maintain stability in vivo, but also give strong expression of the RNA. And you also need a poly-A tail, which all mammalian RNAs, mammalian messenger RNAs have, and you have to have that, otherwise you don't get longevity and you don't get much expression. It also needs to have what's called a cap, which is essentially a small unit of a very short 2 to 3 unit nucleotide, which protects the cap of the, it protects the end of the RNA, but also encourages ribosome binding. And that whole package is required to give you strong expression. But we have methods of making it in vitro. We can add a cap later, or you can add a cap during the process. And then you can purify it. So it's very much like the sort of pharmaceutical product we like, something which we can purify and characterize very well. So it gives us fewer quality assurance problems during manufacture than making, say, a viral system or using an inactivated virus. One of the other important points about mRNA is that because it's RNA and our cells can't make DNA from RNA, then it's very safe in comparison to using a DNA vector. There's no possibility that the RNA can really interact with our genomes. So it can't lead to what's often called insertional mutagenesis in the gene therapy field, which means that a piece of genetic information can't be inserted into our genome and potentially cause harmful mutagenic effects, such as perhaps driving production of a cancer-causing gene. So it's much safer. Very rapid, which we've talked about before. The other thing about mRNA is that because once you've got the mRNA and you've got the delivery system, you have a platform which can be modified very easily. So when the virus mutates, as we're seeing happening now with the coronavirus, that we can adjust the sequence very quickly. And the Moderna and BioNTech Pfizer products are already being modified to produce vaccines against the variants which are of most concern at the moment. Another thing is that if you have some variants, potentially you could have a cocktail of mRNAs which protect against a variety of strains. So we could have a cocktail which contains perhaps the UK strain and the South African strain, which are causing concern at the moment, possibly even the Indian strain as well. We don't know too much about the Indian strain yet, actually. It is different to the South African strain, but whether it's more contagious, we're not quite sure at the moment. I think with the UK strain and the South African strain, there's no doubt that they are more contagious. And scientists are currently looking at the Indian strain to ask that question. If it turns out to be more contagious, and it turns out that the vaccines don't protect particularly well against it, then we'll have to act very quickly and produce vaccines against that strain. Another interesting feature of RNA, which I'll show you a diagram in a moment, is that you can produce something known as self-amplifying RNA, which amplifies the number of molecules produced in the cytoplasm, and means that you can give a lower dose. And giving a lower dose is good for two reasons. One, it means that you can produce more doses per unit mass. So potentially it's easier to vaccinate large populations, but also it means that you can produce a dose which is much smaller, but also it means that the delivery system, which is usually a lipid nanoparticle, is also given in a smaller amount. And those lipid nanoparticles are not particularly well tolerated. They have led to some adverse events with the Moderna and the Pfizer vaccines. And so you can reduce the number of adverse effects, minor adverse effects, but still uncomfortable effects during immediately after the dosing. Another thing that's important to recognize is that the mRNA vaccines that have been approved are not just native RNA. They contain chemically modified nucleotides, specifically the uridine nucleotide. And that has a big effect on the expression and also the innate immune responses to the vaccine. And so modifying the uridine content and modifying the actual chemistry of the uridine component itself is something that can be re-engineered to give you either longer term expression and modify also control, if you like, the innate immune response. You need to have some level of innate immune response to this vaccine, otherwise you won't raise antibodies, you won't raise an adaptive immune response. But you don't want the innate immune response to be too severe. That's the sort of thing that gives rise to the rather uncomfortable adverse events that some people have suffered. This is just a diagram of what the self-amplifying RNA is. So it's actually a fusion of the mRNA, which is shown as gene of interest mRNA here, a fusion to another piece of RNA from a different virus, a virus from a group of viruses known as alpha viruses. These viruses are mRNA viruses that can replicate their RNA in the cytoplasm of cells. So if you graft the replicon part of the alpha virus to your messenger RNA, you end up delivering something which can amplify itself. And so a smaller dose gives rise to lots of molecules of mRNA and lots of protein antigen. And as I said on the previous slide, it's estimated that the dose in a human with this kind of vaccine could be as little as 1 microgram, extremely small dose. Now, at the moment, there are no self-amplifying RNA viruses, self-amplifying vaccines approved for COVID-19, but there's one which is close to being approved, and we might see that it's from a company called Agrecetus in the US, and that is a vaccine which is in the phase 3 of clinical trial at the moment. I thought I'd mention the variants, and this is not really up to date. This is from last year, the middle of last year, but it illustrates the point that there are mutations in the receptor binding domain, which are essentially these ones here, but there are also mutations all through the spike genome, the genetic sequence of the spike. So you can see that these are incidences of mutations that have been recorded and isolated from patients. And you can see that there's loads of mutations potentially that can occur, but probably the ones that are the most important are the ones in the receptor binding domain, because they're the ones likely to affect binding to the receptor. And there's one in particular here which has become almost become the entire pandemic around the world. So this mutation here, where an asparagine, a position 501, is replaced by a tyrosine, This is the UK variant. And the UK variant consists of, all of them have this N501Y, but they might have other variants in different positions on the spike as well. But the one that's critical is this N501Y. Now, the South African variant, which is not really shown on this slide, it emerged later than this, also contains the N501Y, but contains some mutations which were not present at the time this slide was produced. So there's two other mutations in the spike protein, in the RBD part of the spike protein, which are relevant to the South African variant. What we're finding at the moment from the studies in patients, but also the studies in the laboratory, is that the Wuhan vaccines protect quite well against the N501Y variant. So the N501Y actually does bind to the receptor more strongly and is more contagious, but the vaccines do protect against it, but they don't protect very well against the South African variant. They're only about, they're about a tenth as effective against the South African variant as they are against the original Wuhan or the UK strain. And that sort of signals that we're going to have to look at the South African variant more carefully, and perhaps we need to vaccinate against that. And that's what Moderna and BioNTech, Pfizer are doing, producing vaccines with that South African variant. One of the questions that is raised by that, though, is, well, will those vaccines actually work in patients that have already been vaccinated with the Wuhan strain? And that isn't so clear. The danger is that if you vaccinate with whole spike with the Wuhan strain, and then try to boost with a South African variant, you may still raise non-neutralizing antibodies against the original Wuhan spike and not new antibodies against the South African receptor binding domain. So that's a concern, and one which we need to address quite quickly. And that is the sort of thing that people are working on now with a view to making second generation vaccines and making them work as effectively as possible. So this is a piece of data from my colleagues at the Doherty Institute, Damien Purcell's group, and it just shows that the binding affinity, shown in red here, of this is the UK variant, the 501Y. This is a binding isotherm. It binds more strongly than the original Wuhan strain, shown in blue, the 501N. And without going into great detail, I've also shown that convalescent patients who have been infected with the original Wuhan strain, the wild type strain, are not as effective at the convalescent patient's sera, are not as effective at neutralizing the RBD 501Y, the UK strain. So this is a titration curve which represents to what extent can you dilute the serum and still neutralize infection of mammalian cells. And so a high value over here represents good neutralization. So the wild type strain can neutralize quite well against itself, but is not very good against the red strain here, which is the UK strain. So Moderna have already produced a vaccine against the South African strain, which they're calling, their vaccine is called mRNA 1273. The new vaccine is 1273.351 because the South African strain is called B1351. So it just took 30 days for Moderna to adapt their vaccine from the original 1273 to this new vaccine. That's the value of the mRNA platform, that it can immediately pivot and address a new variant very quickly, whereas to do that with a protein or a whole virus would take a lot longer. And the supply is also something which is has been amazingly rapid. So Moderna are talking about producing billions of COVID-19 vaccines in 2022. Obviously we need them, but it's remarkable that a company the size of what Moderna was before COVID-19 can be in a position where it can produce numbers of vaccines that are getting close to providing a global supply. So if we can get a few billion from Moderna and a few billion from Pfizer, add the other vaccines to it as well, we may well have enough vaccines to vaccinate as many people in the world that want to be vaccinated. And that would be quite an incredible achievement. So just to finish off, I thought I'd show you one or two things that are different about mRNA vaccines, which are, this is a bit of technical detail, but quite interesting for those of you who are interested in how to go about making mRNA vaccines. The first one is that I mentioned before that both the Moderna and the BioNTech Pfizer vaccines are using what's called modified RNA, meaning that the base uridine is not the natural sequence. So this is uridine triphosphate, which has the uridine base structure, and native RNA contains this structure here. The pseudo-UTP, as it's called, is a structure which is an isosteric replacement for uridine, but you can see that it's got a nitrogen in a different position in this ring. And that creates a structure which is nothing like as immunogenic as native RNA. And Moderna, the very name Moderna comes from their concept of that company, which is to use modified RNA. BioNTech have used the same approach, but not all companies are using modified RNA. So there's a vaccine being produced by a German company called CureVac, which is close to being approved now, and that is a vaccine made with native RNA. So it will behave differently, and be interesting to see how effective it is in comparison to the Moderna and BioNTech enzymes. So when you look at, actually I'll go back a second, if you look at replacing UTP with pseudo-UTP, it's normally given this symbol. And you can see that in the sequence of mRNA, there are lots of uridines which have been replaced. So it has quite a big effect on the chemistry of the molecule that you're delivering. There are lots of uridine bases this is the BioNTech sequence that they've actually produced. That's part of it, and there's the other bit. And you can see that in bold is this is the genetic sequence that's being expressed. So there's a region upstream which is not in bold here, which is the untranslated region. That's the cap, which needs to be a particular structure for it to work properly. And then there's a long 3' untranslated region, and then here the poly A tail, which has to be pretty long for it to work effectively. In our vaccine, we use about 100, a poly A tail about 100 bases long. This is the cap structure that I was referring to, and it's quite a sophisticated piece of chemistry, this. We buy a cap from a company called TriLink, an American company based in San Diego. And this is a very easy way of making mRNA. If you buy this material called Clean Cap, it basically allows you to add this chemical to the 5' end of the RNA, actually during the transcription process. You need to have that cap to get efficient expression, but this company produces a very nice way of adding that. Makes it very easy for us to do, and without that, I don't think we'd be able to make RNA in our laboratory. You have to make this chemistry yourself, which is obviously a fairly complex piece of chemistry. I guess finally, the thing to mention at the end is that to make an mRNA vaccine, you have to use 3 steps for production. You have to make some DNA, which is made in E. coli, purify the DNA. Then you have to go through that mRNA transcription process. But then you have to assemble it into a formulation. And the formulation is actually the area where most of the new patentable intellectual property lies. So the companies that make mRNA vaccines have slightly different ways of assembling their mRNA into a particulate form. They're all lipid nanoparticles, but the lipids that make up the structure are slightly different. And this is an area where there's a lot of scope for research and also a lot of scope for new intellectual property. I'll just briefly show you what's involved in making the formulation. It's a strange formulation, but one which works well, and most of the companies use a similar sort of approach. What you have to do is dissolve your messenger RNA in an aqueous buffer, and then you have 4 lipids, typically this kind of group of lipids here, dissolved in ethanol. And you mix up the ethanol solution with the aqueous buffer solution containing the mRNA. It gets mixed in a device and then it gets dialyzed, and then you produce particles of around 100 nanometers with the mRNA trapped inside them. The reason the mRNA gets trapped is because one of the main components is cationic. So in this example, with these 4 lipids, this particular lipid is a weakly basic lipid, can be protonated, and when it's protonated at pH 4, because you tend to make these things at pH 4, it obviously binds quite strongly to the negatively charged RNA and draws the RNA into the particle. So this is a critical part of the formulation, and it's 50% of the lipid is made up of one of these ionizable lipids. There's also a lot of cholesterol. So cholesterol is obviously present in biological membranes, and cholesterol is in all of the formulations that are approved for human use. So it seems like the cholesterol is an important part of causing the LNP molecule to bind to mammalian cells and get taken up. And cholesterol is well recognized by our mammalian cell membranes. And I don't think I've seen a formulation which works, which does not contain cholesterol. So that's an important thing to think about. And then there's also a couple of other lipids, one of which is a natural helper lipid, a natural neutral lipid, and then there's a pegylated lipid, which is there only in a small proportion, 1.5% typically. And that just allows the surface of the particle to be coated with PEG and allows you to make nice particles which are stable. So this is very much a pharmaceutical, these are pharmaceutical agents, and this is a rather strange and wonderful pharmaceutical formulation. As I mentioned, that's what we're using at the moment, but there's quite a lot of scope for improvement. And I think with that, it's probably time for me to close the talk off, and I'm very happy to answer any questions if you have any. So I'll stop there.
Hello, 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 Professor Colin, can you hear me?
Yes, I can, yes.
Oh, you're wonderful, awesome lecture, Professor Colin. Thank you so much. It's so comprehensive and very clear and well delivered. Thank you so much. And so this is the time for our question and answers. So we will now, Gayatri is there. So Gayatri, you can just entertain certain questions of interest.
Yes.
Gayatri.
So yeah, so if you have any questions for Professor Pouton, so feel free to unmute or type your question in the chat box so we can take the questions
Yes.
now.
And Professor Colin, I do have a question. You know, you were talking about these mRNA vaccine formulations. I'm talking about these the last few slides. In the last slide, you were talking about the like formulations of this mRNA with lipid nanoparticles and lipid molecules. And so you said the lipid has to have a cationic in nature to anchor with mRNA moieties, and
Yes.
so at pH 4 it is there. But how about when you administer it into the body? The pH of the body is different. So what will happen? What will be the fate for these the particles?
Yes, I probably didn't make that very clear, Rohini, because they actually make them at pH 4 initially, and it's necessary to do it at pH 4 to get the mRNA encapsulated. But then they actually do dialyze the buffer to get to pH 7.4, and that I probably should have mentioned that, but it does involve it must involve a substantial rearrangement of the morphology of the particles, but and it's not a very well understood process actually.
Okay.
In the end, the particle is not really cationic. So once it's raised to pH 7, I probably should have said the pKa of most of the lipids used is around 6.5. So they actually go from being protonated to unprotonated when they dialyze them. And it probably means that there's a huge structural rearrangement that occurs during that process. And in the end, it's like a blob of unprotonated lipid, which has RNA trapped inside it. So it's very unusual, actually. It's not really like a liposome at all. It's something different. It's like a big lump of lipid rather than a liposome.
Right. Okay. Very clear. Thank you. Yes.
Yes, Professor. Yeah, Professor Pouton, I have a few questions too. So, do you think that adding additional components like conserved end proteins of the coronavirus may perhaps optimize the efficacy of these mRNA vaccines? And my second question is, well, I have learned that COVID-19 prevents certain people from forming germinal centers. But there's this new information says that this LNP mRNA vaccines show very efficient induction of these germinal centers, well, at least in mice. So in that case, do you think that the vaccination may outperform natural immunization?
Yeah, look, it's so early. I don't think we really know all the effects of these vaccines yet and how they differ from one to another. It's one of the most humbling things about this whole process that you realize that now we're vaccinating huge proportions of the world's population with something which we don't fully understand. I mean, it's very much, there's so much to learn. I guess we'll continue working on both the virus itself and the effects of these vaccines for many years to come. And really, like you say, there may be a need for much more sophisticated ways. Right now, we're doing the most obvious thing, which is just to try and raise antibodies against the spike. But there's all the other proteins as well. And they may be doing some very complex things to our cells that we don't really understand.
All right. So there's a question in the chat box. Are viral immunogens that induce neutralizing antibodies identified with those that induce non-neutralizing ones?
So the question is, have we managed to identify which antibodies? Is that the question?
Professor Pouton, it's not quite clear for me also.
Okay, well, I think what I'd say about that is that because it's not really an easy thing to do, but because there are big groups working on the science of these vaccines, they have isolated a lot of different antibodies and tested which ones are genuinely neutralizing and which ones are not. And one of the things we know, I suppose this is common knowledge really with vaccines, but it's certainly happening with the COVID vaccines, is that during during the development of the response, we get a maturation towards biasing the immune system to produce antibodies that are actually neutralizing. So particularly if you get COVID-19, rather if you get infection, I suppose COVID-19 is a bad form of the disease, but if you get infection with the virus, you get initially production of a whole variety of antibodies against the spike, but then it matures into the most useful populations in an individual that has a good immune response. And the same thing happens with the vaccines when we do experiments in mice, that if you look after the first dose, you'll see that you get quite a high titer of antibodies, but the number of antibodies within that pool that are genuinely neutralizing is not as great as after the second dose. So it's very interesting part of vaccination. You probably do need a boost, if only to help the maturation of the immune system. So we find that after the boost, the number of antibodies produced, the whole antibody titer is not really very much bigger. It is bigger, but not hugely bigger, but it's certainly much more effective at neutralization. So this is something which is obviously part of the normal function of the immune system. And it's probably why we do need more than one dose of a lot of vaccines to help that process along.
Right. So related to that, one person is asking, so if a person gets the second booster of the Covishield vaccine, is it okay to get the China Sinopharm after that? So,
yeah, that's a very good question. And I would say that in principle, there doesn't seem to be any reason why you shouldn't have a boost with a different vaccine. Initially, actually, when the UK started vaccinating, because they were one of the earliest nations to start vaccinating seriously, they actually realized that it might be difficult to find the second dose of each vaccine to vaccinate people, and they were thinking of mixing them up. And they actually got a lot of criticism from the WHO at the time, because obviously when the vaccines are first approved, it's the result of a clinical trial, and that clinical trial is with the same vaccine, a prime and boost with the same vaccine. So to actually say straight away that you're going to mix and match vaccines is sort of going against the data that the trial has been used to approve the vaccines. So they actually, it was frowned upon. But on the other hand, now, as a little bit later in the piece, they have actually been doing that. Not in a widespread way in the UK, but they have been, in some cases, testing in a trial, in a sort of formal trial. They've been testing priming with one vaccine and boosting with a different one. And they've found that it works very well. So I guess that will be used as evidence to say, well, yes, it is okay to prime with one vaccine and boost with another. And I suppose if there's an acceptance of that, it'll make it much more practically possible for people to get two doses.
exactly, right.
But one of the things to say about that, which is also very relevant, is that actually the adenoviral vaccines really don't work particularly well on the second dose. And that's because you raise antibodies against the adenoviral vector itself. So the AstraZeneca vaccine, the Oxford AstraZeneca vaccine, as well as the J&J vaccine, really are principally depending on the first dose for the vaccine effect. And the second dose, if you look at the results of the clinical trial for the AstraZeneca vaccine, the second dose is not really that much of an advantage.
Okay.
And that's why J&J have made their vaccine a single dose vaccine. Interestingly, on top of that, the Russian vaccine, so-called Sputnik vaccine, is actually two different adenoviruses, two different serotypes of adenovirus. And so that actually works better because you give you give a boost with a different serotype of adenovirus and you get a much better response.
Yeah.
So that's an interesting aspect of all of this.
Yeah.
It kind of tells me that probably if you've had the AstraZeneca vaccine, you're probably better having an mRNA vaccine boost in reality. But I don't know whether that's practically possible at the moment, but it's probably the reality.
Okay, there's another question. So which type of vaccine would be more efficient for the people who have already got COVID-19? So
yeah, it's a very good question. I would say that probably the vaccine, the type of vaccine we're working on, which is an RBD alone vaccine, would be a better way of approaching it. But actually, there aren't any available at the moment. But I think they'll come through because I know there are RBD vaccines in development. So, yeah.
And one person is asking why this CoV-2 undergoes rapid mutations compared to other virus.
Yeah, that's another good question. Well, I think it's not particularly special to coronaviruses or CoV-2. I think it's just the RNA viruses all have a tendency to mutate. And that's because they can't use host cell machinery to replicate. So RNA viruses, we don't really have RNA replication system for which the virus can adopt. And so they have to bring their own enzymes into the cell. And those enzymes are not very high fidelity. So they tend to make mistakes. And that is part of the viral life cycle, I suppose. It brings its own RNA polymerase, and that itself is prone to error. And so it causes a lot of mutations as a matter of course. So DNA viruses don't mutate as much. And that's because they use host cell machinery for replication.
Okay, so student is asking, would giving an mRNA cocktail have adverse side effects if non-neutralizing antibodies are produced?
Yeah, we don't. Well, no one's really using one at the moment. It's more of a hypothetical suggestion that we could use a cocktail, but no one's actually used a cocktail yet.
Right.
It might be that we start to use a cocktail in the future, but one of the things is that the technology is slightly behind the pandemic, if you see what I mean, because by the time we start to use variant vaccines, quite a lot of people will already have been vaccinated with the Wuhan spike. So you then have to ask the question, do you use a different approach for people who've already received a Wuhan spike to someone who's naive and could potentially have the South African vaccine? And I think those questions just haven't really been asked yet.
Right.
I think we don't really know at the moment how many of the doses will become South African vaccines. They've just produced them and they haven't actually been approved yet for human use. I'm sure they will be soon, but how many of them are going to be produced? I'm not sure. We'll have to wait and see.
So
And then,
hi, Professor Colin. It's Malindra here from Astra Limited. I have seen some research done on some mouthwashes and all that from UK as well as some other countries as well. What is your perspective on that?
so I didn't quite catch the question, Malindra. Can you just say what you said again?
I have seen some research done on mouthwashes and stuff like that from external washers.
Oh, okay.
Yeah.
Do you mean a way of administering the vaccine in that way?
No, not vaccine. What I was thinking is how can it be destroyed like that?
Oh, the virus.
Yeah.
Yeah. Well, obviously, there's all sorts of approaches to trying to reduce the viral burden. And it makes sense to me that if you've got patients who are really sick, so patients who are getting hospitalized with a serious case of COVID-19, under those circumstances, when they're in, particularly those who end up with severe breathing problems and so on, we probably do need to have interventions that can be used for those severely ill patients. And there's a whole load of different approaches to that which are being developed. I'm not really particularly knowledgeable about them, to be honest, Malindra, but it seems to me that there is a need for antiviral treatments for those patients who get really sick. And we hope that some will emerge that are useful. Unfortunately, the drug treatments that people have used in the during 2020 didn't really seem to help much at all. But maybe some more targeted approaches to maybe shut off the viral replication will help. Some people are using genetic approaches now, like RNA inhibition or even CRISPR type approaches to try and stop the virus replicating. But they haven't been tested in humans yet.
Okay, thanks very much.
Yeah, Professor Darling, go ahead and ask your question.
I think that's Gayatri, there's a question from Tilan also. Can you ask that first? He posted before you.
Yes, so he was asking whether there's any effect of these vaccines on people who already recovered from COVID. Are they better off not getting the vaccine?
Yeah, that's another very interesting question. And I think most people who have had a mild infection are getting boosted at the moment. In countries where they have a lot of vaccines being applied, like the UK and in the US, they are immunizing patients who had a mild form of disease. I suppose because at the moment, the serum samples from convalescent patients are they don't have as many antibodies as patients who've been vaccinated. So probably it is worth vaccinating those patients, particularly if they're aging patients. But once again, you wonder what's the best vaccine to go with. I would be saying that probably in the long run, it will probably be an RBD-based vaccine rather than a whole spike.
Yep, so I have another
Thank you,
question.
Prof.
Yeah, go ahead.
Okay, Gayatri?
Yeah, so
Shall I ask?
Yes, go ahead.
Okay, Professor Pouton, thank you very much for your wonderful presentation. I'm Professor Nalin, Head Chemistry in Colombo. And I have a question. I'm a material scientist. I'm not very conversant with the technology that you are talking about, but I have this question regarding the luciferase is used in Moderna, because I know the luciferase is a enzyme that is used up in Firefly to convert the luciferin to give light. But what is the truth behind that the luciferase is used in Moderna, and also are there any effects on that, or what's the purpose of luciferase, or it's a complete lie? What can you comment on that? Yeah.
Well, there is no luciferase gene in the vaccine itself. Probably Moderna would have used luciferase in the development of vaccines. A lot of people do to effectively to test how the quality of the delivery system. So it's a good way of, it's what we call a reporter gene. So it's something that we can easily measure. Luciferase, using luciferase rather than say a SARS-CoV-2 sequence means that you can measure the effectiveness of your vaccine delivery much more easily. So it's used mainly to distinguish between delivery systems in the development of delivery systems, but you wouldn't find it won't be found in the Moderna vaccine itself, no.
Okay, thank you very much.
Okay, so there's another question from me actually. So is there any relationship with the age and this mRNA vaccine associated side effects, Professor Pouton?
Yeah, I don't really know about that. At the moment, there doesn't seem to be an age difference. I think what's happening is that some people are getting the minor side effects at the site of injection. So they're getting pain, at the injection. Sometimes they're getting headaches, sometimes they're getting a bit of fever, but it doesn't seem to be age-related. And then some patients have had, and you may remember early on in the when the vaccines were first started, there were some instances of anaphylaxis, rare instances, and I guess that is something that would happen potentially with any injection, certainly of a protein. So any vaccine, you'll get some anaphylaxis, but the sort of just the discomfort which comes and goes within a couple of days, that doesn't seem to be age-related. It may be something to do with whether or not patients have seen coronaviruses, because it's possible that they're raising some sort of immune response locally against some of the components they're being vaccinated with. But I think it's hard to know whether it's just a non-specific effect or something directly to do with the coronavirus spike. The same thing goes for the clotting, which we've heard a lot about. The clotting issues seem to be actually related to the viral spike protein itself, and so potentially can occur with any of the vaccines. And in fact, there's quite a lot of evidence now that if you get COVID-19, you also potentially can have these rare clotting effects, but you're much more likely to get them if you have COVID-19 than if you just receive the vaccine. So that's another interesting thing. That's actually probably to do with the virus rather than the vaccine delivery system.
Yes, so we have limited time. Unfortunately, even though we have tons of questions in the chat box, we have to wind up.
Let me.
Yes.
Yes, Gayatri. Yes, as Gayatri says, it's so interesting and it's a timely topic. So there are thousands of questions, and we can keep you here for an entire day. I'm so sorry because you have given us the limited time, and you are a very busy person. And thank you ever so much. And we will continue to be touching with you. And so now, I have to thank Amali, my student, as well. And to introducing you, and also the particular webinar is sponsored by Astron Limited. Thank you for them as well. So now it is the time for the official and formal thank you speech, and it will be delivered by Dr. Gayatri, and she's the secretary to the A2 section. Thank you very much, Professor Colin, again. Okay, over to you, Gayatri.
Thanks, Professor Rohini. So hello, everyone. It's the time to draw our session to a close. So thank you so much, Professor Pouton, for bringing your perspective on these highly efficacious mRNA vaccines and sharing your expertise on vaccine development against this SARS-CoV-2. That was such a timely presentation. And it's truly remarkable to see the rapid evolution of these novel and effective vaccines for COVID-2 in less than a year. So on behalf of SLAAS E2 2021 team, we wish you all the very best to drive your research to the next dimension in this new year to successfully combat COVID-19 pandemic with your timely innovations. So thanks again for honoring us with your presence and educating us on mRNA vaccines. And I would also like to extend my gratitude to our event sponsor, Astron Limited, and Amali Guruge for arranging this fantastic talk. And I'm also thankful to the students, academics, and researchers for your participation, response, and cooperation to this event. Thank you for such productive discussions, and thank you very much for joining with us. And last but not least, I would like to extend my gratitude to all the people who directly and indirectly helped me to make this event successful. And all these webinars organized by SLAAS E2 and our past sessions will be available on SLAAS webpage. So we will be also announcing our future webinars on the same page. So please visit www.slaas.lk for more information. And thanks again, Professor Pouton. And over to you, Professor Rohini.
Okay, thank you so much, Professor Pouton. Thank you so much. Bye-bye.
It's a pleasure.
Bye-bye.
Thank you for attending.
So honored to have you.
Everyone.
Yes, thank you so much. It's so productive.